M42, the main stage, the nexus between technology, sustainability and health. We've already kicked off with fantastic thought leadership presentations, thought-provoking panel discussions, and some riveting presentations as well. The next panel discussion really builds on this and adds to that knowledge exchange. I am delighted to introduce the topic area that we'll be diving into, harnessing the power of proteomics, which is a new medical innovation. I've just been learning about it off stage, so the speakers are really exciting to share everything with you. Without further ado, allow me to introduce the speakers. Dr. Tiago Magiage, the Director of Bioinformatics at M42. Please do welcome him on stage. Dr. Nicole Sirotin, the former chair of preventative medicine and consultant internal medicine at Cleveland Clinic, Abu Dhabi. Dr. Sarah Solomon, the consultant endocrinologist and clinical lead for genetics at the ICLDC. And Dr. Shalini Bahal, the R&D scientist, Omics Center of Excellence for M42. And I'm literally handing over my mic to Albara Elkani, the Senior Director, Operation M42, and he's just going to make sure that everybody is seated in the right place. Over to you, Albara, and thank you so much.
Good afternoon. Is that the right seating? Can we confirm? Yeah, okay, very good. It's very confusing. Thank you. Good morning, all, or good afternoon. So I think I'd like to set the stage a bit for proteomics. There is a new school of thought within the medical domain, what we call the translation of our genomic blueprint. So the genome is our blueprint that describes our functions, our physiology, but there's a translation of that into proteins. And this is where we're going to be describing the protein as a technology and the potential applications within the clinical domain and the research domain. So before we get into sort of the panel discussion, I'd like the panel to introduce themselves and how they specifically have worked in this domain, if you don't mind. Dr. Sarah, we'll start with you.
Good afternoon. Hi, my name is Sarah Solomon. I'm a consultant endocrinologist and diabetologist. I lead the genetics service at Imperial College London Diabetes Centre, having done my PhD in Oxford in genetics. Proteomics is one of these new things, and we'll speak about it in a minute. Why don't we finish the introductions, and then I'll come back and say why I feel it's relevant to my care.
Hi, my name is Tiago Magalhães, and I am the Director of Bioinformatics. And at M42, we now have a Proteomics lab, and my team is responsible for analyzing and doing the bioinformatics work on that.
Hi, I'm Dr. Shalini Behel, and
Mike,
I'm
a
a
bit
research.
Closer.
Hi, I'm Dr. Shalini Behel, and I am a research scientist at Omics Center of Excellence, mostly into the Marathi genome program, and now moving on to proteomics.
Thank you. Good afternoon, everyone. My name is Nicole Sirotin. I am a consultant in internal medicine and have been a physician leader as a department chair for the last 10 years at Cleveland Clinic, Abu Dhabi. Have been there from the beginning of our project and watched it grow into this amazing hospital that it is, and have been really focused on bringing innovative programs and technology through the Cleveland Clinic Abu Dhabi system and now M42 to the UAE.
Thank you so much. They're actually being extremely humble. They're trailblazing in their field, to be fair, and this combination of experts we have on the panel today is a very unique situation. I'm very fortunate to be here today. So, Dr. Sarah, question for you. We know proteomics from a scientific perspective, but can you please explain to the wider audience here, what does proteomics mean and how that applies to our sort of body and our health and well-being in general?
Thank you very much for that question. I'm a simple person, so I like to put things in simple words. For years, we've been focusing on the genomics, on the DNA, and that is what the architect gives you. If you think in architecture terms, it's a blueprint, it's on paper. That then is transcribed and RNA is produced, and that is the building. So all the walls, all the doors, everything else. And proteomics, to me, is the furniture. It's what makes everything work, but it's quite mobile. So as we heard from Matthew earlier today, his wife keeps putting things in different rooms. And the beauty of that, it's almost the final destination of what comes from the DNA. So rather than looking at a DNA which we have from the day that we're born, and if you think about it, DNA tells us an awful lot, but it also doesn't tell us an awful lot because we have an eye, we have a nose, we have a hand. They're not the same. What's happened to that DNA is alterations in the DNA for it to be transcribed. And then the RNA also has alterations. Some of it will break down and never make a protein. Some of it will end up as a protein. And actually, there is a further step to proteomics, which is the proteins that are made. Some of them will have alterations such as acetylation, glycosylation, methylation, which will make it function better. But that is the end point. These are the things that are actually making the difference. They're the hormones that make everything work. They're the enzymes that digest our food. They're part of our cell structure. So we're looking at the bottom of that. And the beauty of that is because it changes, we can look at markers that will tell us they're related to a particular disease or what medication might be able to work. It's a really strong tool. And I'll leave it to my colleagues to go into how they technically do that.
Thank you, Dr. Sarah. I think that summarizes proteomics very beautifully in a simple manner. You know, it's the building blocks that allow us to translate our genomics into what we have today. So, Dr. Nicole, does the environmental factors affect your proteomics? And can you explain a bit more about that?
Yes, thank you very much, Dr. Sarah, for your beautiful metaphor. I think if we think about the house and the building and the furniture, what happens in that room really matters. And so, for example, if you were to come into that room and, let's say, have a very big feast, and that feast included lots of fatty foods and lots of high caloric foods, there might be a little bit of mess to clean up, let's say, in that room. And those proteins will change based on, let's say, the food that you're eating, because the food that you're eating then has a cascade of signals in your body. Or, for example, how much you're moving your body, right? And so the proteins really are adapted both to what you're exposed to. So, for example, if there were environmental toxins in that room, the furniture might be changed slightly from what it's exposed to. And then the way that you live your life, how you eat, how you sleep, how you move, how you stress, all of these have biochemical pathways that then change the protein expression. For example, fasting is a great one because we have very nice studies looking at caloric restriction, so going without food, and then looking at the way that your proteome or your protein patterns change. And so we can see very clearly in studies how before and after a fast, you might have a different pattern of proteins. So you could say, how does that help us? Well, what we can see is patterns that are associated with certain risk or certain diseases, and then patterns that change with an intervention, whether that's a medication or a lifestyle intervention.
Thank you so much. I think also for the audience who have an understanding of proteomics, Dr. Tiago, a question for you. Proteomics is actually not a new technology or innovation. It's been around for years, right, as a methodology. So what has changed in the last few years that is bringing proteomics to the clinical forefront? What innovations has happened? Can you explain more about the development of this product?
Yeah, I mean, like the audience is saying, you have DNA, you have RNA, and you have proteins, right? And since '56, I think, when DNA was discovered, people are trying to get information about DNA, about RNA, and about proteins, right? But of course, before it was very troublesome I remember kind of you had the gel was by weight, then you apply something, electromagnetic current, these are electric points, and you had this blurred, very weird kind of little dots, and each one was a protein, and you had kind of to guess. But that was 300 proteins, right? Very, very few. Now we have 7,000. So suddenly you try to see patterns. And like Dr. Nicole was saying, intervention, you can see the changes, right? Imagine there is an intervention that incredible study you did on caloric restriction, right? People didn't have caloric restriction. They have a set of proteins. Then there is a caloric restriction. They have another set of proteins. You compare the two, you have 7,000. You don't know what's happening with 7,000, but now we know. The ones that are changing, you can say that's a pattern. One, you can read that pattern and see this is what happens when people are caloric restrictions, and you can also intervene. Because maybe you find one gene or one protein where it's going up, and maybe someone says, okay, if this is happening on caloric restriction, how can I intervene there with a drug or some other method where that's changing, and then you mimic the caloric restrictions. And that's the power of going from 300 to 7,000, and many more will come, where if suddenly you have the full picture of what's happening in the cell. That's the power of this.
So if I was to summarize, what we're saying here is the correlation of the protein biomarker changes based on stimuli, clinical condition, is giving us an insight to help us predict a condition prior to happening.
For instance,
clinically.
yes. And also how to intervene. For instance, long COVID. Last week, paper on Science, SomaLogic, all these panels, they understood what are the genes that are changing long COVID. When they can check, is this long COVID? Because now we know what the proteins are changing, and they can also intervene. So that's the power that this gives you. It gives you all the understanding of what's happening in the cells.
Perfect. Thank you, Dr. Tiago. Dr. Cellini, so you're a researcher within M42, right? How have you found the protein assays, this 7,000 proteins and 10,000 protein biomarkers? How have we seen the research applications of this, and then bringing it back into the clinical field? Can you draw us a picture of that, please?
Substantial research efforts have been ongoing at the Omics Center of Excellence at M42. And the technology that we are currently using to detect all of these protein biomarkers is we can detect 7,000 biomarkers, which are currently we can build algorithms for cardiovascular diseases. There is body fat, glucose tolerance, alcohol consumption tolerance, and so on. One study that we have recently concluded is on a cohort of participants with risk to cardiovascular events and some control samples. And we concluded a 4-year predictive model where we could rank them from low to high risk. So I believe this is not only restricted to cardiovascular events, but could be extended to other clinical fields like oncology and diabetes, etc. So with this in mind, I also take this opportunity to invite collaborators, experts and researchers here. So we could build a database where we can have predictive models for using understanding protein dynamics and also using them for research and building predictive models.
Thank you. Maybe just to also add a twist to this as well, from the research that's been done both by the manufacturers of protein kits and by the clinicians, there's a statement that's made for specific types of disease burdens, but you can predict a potential cardiovascular incident, myocardial infarction for example, 4 years prior to happening, just by looking at the protein makeup. So these are very powerful tools that we are just yet to sort of scratch the surface of. So based on the sort of clinical applications, maybe Dr. Sara, if you can just give us a bit more. I know that you're very heavily involved in the diabetes link and the cardiovascular disease and the protein markers. If you can give us an understanding of the clinical applications and where you see proteomics being used in the clinical domain.
So I think proteomics is the new frontier. It's where we're heading, it's where the future is. We have had proteomics for a very long time. So for example, in prostate cancer, we've had a PSA, single protein, and we've tracked it. It's told us if the disease has gone away or the disease has come back. It's amazing. It's targeted treatment. It's helped us. What I see for diabetes, endocrinology, and obesity is that now we can measure things blindly. So it's hypothesis generating. We're giving samples, for example, before bariatric surgery and after bariatric surgery to understand what happens to that person. Why don't they immediately gain the weight that they had lost? What keeps some people metabolically normal, whereas others will regain the diabetes and the weight? So that, I think, is the strength of proteomics. That will help in diagnosing that patient so we can target them with earlier treatment. It will help with prognosis. As we heard from Al-Barah, are they going to have a heart attack? Do we need to really attack everything we can change? It will help with prevention. So if we see people with markers, children, for example, with markers that they are likely to be unhealthy in later ages, we can then even introduce dietary measures, exercise measures at a very young age and track the changes that happen. And ultimately, it's looking at medication, because if we know that that is the protein that's altered, we can then plan a medication that works on that protein. It's very focused. It doesn't expose people to things that they don't necessarily need.
Excellent. Dr. Nicole, I know in a certain capacity you did at Cleveland Clinic, there was sort of an executive health. What do you think the use cases would be for well-being and lifestyle linked to proteomics?
Thank you for that. I think this is a very exciting area for proteomics because on the one hand, everyone wants to know what their risk is. And we have some traditional markers to help you understand that risk. But there are things that we still don't understand, let's say about how inflammation might affect your risk of heart disease. And we know that those are connected. And we have one or two traditional blood markers we might use to check that. But something like a proteomic assay that might look at 10 different proteins that are involved with cardiovascular risk, many of those are inflammatory markers. So that's actually looking at your immune system and not necessarily the traditional marker like a cholesterol LDL level. And so here we can really round out what that risk profile looks like. So if you're coming in and you say, I really want to know where am I on the map of my health, these kind of tests will help pinpoint that more clearly. We did a study actually with this proteomic assay, taking 12 unhealthy Americans, and they flew to Abu Dhabi as part of a reality TV show, and we did these proteomic assays pre and post. And what's interesting is that this intervention that they went through was a dietary intervention. It was a very controlled environment where they were fed all the food that they ate. They were fed. It was completely controlled in a healthy way with a plant-based diet, and we could see those changes. And so the other piece of this that I think is important for clinical practice is to help reinforce positive ways in which your body is improving after an intervention. And so those of us that have really tried to help our patients think about, empower them with tools that they know will help improve their health, the proteomics provides an opportunity to give them evidence, to show it, right? To say, look, this is how you're improving. We can see it in your proteins. Because the proteins change with what you put in that room and where you put your furniture.
It's quite interesting. I would like to see that study of the reality TV show of the Americans. I feel there's a lot of data being generated, right? So we described before the genomic data. We have all of the clinical records sitting within our EMRs for our patients, and the lifestyle data, and now the proteomic data. Dr. Tiago, is there something that we can do using bioinformatics, AI modeling, to expedite maybe the correlation and the innovation and the insights that we can build for proteomics?
Yes.
That's it? Yes? Okay,
Yeah,
Dr. Sara wanted to answer this as well.
That was the question is yes, you can do a lot. And of course, when you think about this, again, you're talking DNA is going to be very relevant. Transcription is going to be very well relevant. Proteomics is going to be very relevant. Methylation is going to be very relevant. Metabolomics is going to be very relevant. And then you start to understand the cells, right? You have many pathways in the cells. You prime one cell, goes to another way. The other way, oh, this is too much, it primes back. And then the other one say, oh, this is something happened, then it primes back. If you have disease, it goes the other way around. And you cannot do that by one simple marker. And this is what we are doing there for many years, right? You go to PSA, BNP, I don't know, those markers, cholesterol, and you say, oh, you have that. That is just not how biology works. So we are missing a lot. I mean, those are very important markers, that's why they work. But when you start adding all this other information, we are going to understand disease, we are going to understand aging, we are going to understand normal feedbacks, and that's when you put it together. But yes, you need patterns, you need computers that learn these patterns because deterministic algorithms are not going to work. And so you need a lot of people, and you need, which is incredible in this panel, you need the cooperation of many different people, right? Right now it's not just about bioinformaticians or clinicians, it's a wide group of people coming together, and I guess that's why M42 is so powerful, is that we have all this together that could bring the power of all these technologies to push.
So
forward.
yes.
Yes.
Okay. Now I know, Dr. Sara.
Now you're complaining that I am taking too long. I don't know.
Well, now you keep talking, so we have to continue. No, Dr. Sara, I know you're very passionate about this as well. So if you can give your insights, please.
Actually, I was going to tell you that Tiago and his team are worth their weight in gold. And if I give you an example, if you're driving, you will know what speed you were driving at. But actually, it matters much more what state of the road is, what other drivers are there, what the speed limit is, it's everything else. And what Tiago and his team do is systems biology. They don't look at one protein. They look at the bigger picture. And with bioinformatics and really powerful computers, they're able to actually put it all together and say, actually, there really is an issue there. Or that's probably an error. It doesn't make sense because it doesn't fit with everything else. And therefore, it's made proteomics that much more understandable, that much more meaningful. So thank you.
Perfect. Thank you so much. I see the love on the stage here, so I'm very happy with that. No, but I think just because we're running out of time, maybe just as a final statement, I would like to hear from each of the panelists very briefly, what they think the most impactful thing we can do in 2024 with proteomics on the life of our citizens in residence here in the UAE. Dr. Sara, please.
The largest killer in the world is cardiovascular disease. Diabetes and obesity both predispose to cardiovascular disease. I would love it if we had a chip that could tell me who was going to run into trouble so I could optimize their treatment and prioritize them.
Thank you. Dr. Tiago.
Yeah, I think that would be my choice too. Of course, cancer, because it's a disease, is a clonal disease, is a disease of changes, and proteomics is about changes, it's about getting the change quickly and understanding quickly. Could be another one, but I think those would be my two shots.
Dr. Shalini?
I will add to Tiago's work here. A very recent study from the Ukraine Biobank was done on solid tumor cancers, which was just published this month, and it was done for just 440 individuals. So we will be happy to extend that study and improve our understanding on protein dynamics.
Thank you. And Dr. Nicole, please.
I will echo Dr. Sara here. We have a very young and sick population in the UAE, and we diagnose heart disease 10 years earlier than North America. We have 45% of all cancers diagnosed under age 50. So we have to start earlier in our population doing screening and risk stratification. And so I would love to see a more accurate risk stratification tool that represents the biology of the people that live in the UAE.
That's actually very important. It's the precise way of dealing with our population here. And I think this is a very important thing. And I think combined here with the community, we can really do a lot of work on looking at our local population, their biology, and the impact of proteomics. I think we have time before we conclude for maybe 1 or 2 questions. So from the audience, is there any questions for our esteemed panelists?
Thank you very much for the exciting discussion. I'm just asking about the use of the power of proteomics in terms of the use of the antibiotics. Can we use it? Especially there is exceeding overuse of antibiotics in the region.
So that's an excellent question. If we do have the proteomic data, we're able to know what targeted treatment we can use on the microbe, but also on the person to see whether it's going to give them side effects or not. So you have the beauty of both sides. Is this something that we can use en masse? No, because your proteome is different to mine, but it is a fantastic question.
Apologize, we have run out of time now, so I'd like to again thank our esteemed colleagues on the panel here. Please, a round of applause for all the team. Thank you.